Welcome to the video on protein, and this is the second of the 4 macromolecules that we'll discuss. So let's first take a look at the functions of proteins. Looking at the various functions of proteins here, you can see that proteins are essential for life, and it is necessary for structural support, for enzymes, and storage, for transport of material in our body. They're also known to be important for being embedded in the cell membrane. So that's also for transport too of material in and out of the cell, but also as receptor proteins that an immune cell wants to make sure that it can recognize our cells from foreign cells. In order for it to do that, it needs to bind to certain receptor proteins. That's just one example. Defense proteins are also essential, and this is part of the immune system. So the immune system works by being composed of proteins. And also there are hormonal proteins or hormone proteins that are essential for signaling. We will talk about structural support and enzymes. We'll talk about all of these in detail during various parts of the semester. So let's take a look at the monomer of proteins. So the monomer of proteins is called amino acid. Taking a look at this particular structure, we can see immediately that it differs greatly when you compare it to the monomer of carbohydrates, which would be monosaccharides. What we see in amino acids is several things. We see a central carbon, and then in that central carbon, we see a carboxyl group that binds to the central carbon and an amino group that also binds to the central carbon. So the difference between these two groups, or the properties, I should say, between these two groups give the amino acids its specific characteristics. And if you were wondering why it's called an amino acid, well, it's called an amino acid because it has this amino group here. And it's called an acid because whenever you see this group, carboxyl groups, that tends to have acidic properties associated with it. So that's why it's called an amino acid. Amino acids also have an R group, or a residual group. That's what the R stands for. And this R is basically a constant, that there would be many side chains that link to this particular central carbon, but that these side chains differ depending on the amino acid. And the R just represents the various side chains that can bind to the central carbon. And so let's take a look at the 20 amino acids that make up our own system. So all of our proteins will need at one point or another these 20 amino acids. I know your book says that there are over 20 amino acids, and that indeed is true, but for our purposes and for eukaryotes, there are 20 amino acids that are essential. And if you look carefully at these 20 amino acids, what you notice is that they all have similarities and they all are different. So what makes them so similar is that they each have, well, actually this would be a really great time to pause the video and I'll ask you if you can pick out the similarities and the differences. So take a moment and do that and we'll come back. Okay, I hope you had a chance to look at these amino acids, and they all are similar because they have the central carbon and that skeleton of having the amino group and the carboxyl group. So all amino acids have this basic structure. But how they differ from each other is that their R group differs. So the side chain differs dramatically, or it can differ dramatically, and these side chains have huge effects on the overall architecture and function of the protein. And if you look carefully at these side chains, you notice that they fall under it being polar versus nonpolar, or something called charged side chains. And anything that has a lot of hydrogen and a lot of carbons has non-polarity associated with it. So a lot of carbons and hydrogens attached to it is non-polar. So you can see immediately that the side chains that have carbon and hydrogen are the side chains that also are non-polar. So let's take a look at this reaction of building a polypeptide, or we're building a protein. So we have 2 amino acids here, and we want to create something larger. We want to create maybe 2 or 3, and we create a chain of amino acids. So how can we do that? Well, this is the same idea of going back to that very first video on macromolecules. Whenever we're building something, this reaction is called a dehydration reaction or dehydration synthesis reaction. So we're taking one amino acid and taking another amino acid. We're taking away water from the system, and notice that the carboxyl group of one amino acid will interact with the amino group with the other amino acid. And this bond that forms is called a peptide bond. So the bond that forms between amino acids is called a peptide bond. So if I were to build, we have these amino acids, and each amino acids have a particular symbol associated with it, and oftentimes they are these 3 letters, and those 3 letters represent the first 3 letters of the amino acid. So in this case, we have methionine, MET. So if I were to take methionine and I wanted to link it using this kind of a reaction with another amino acid, like glycine, I'd get something that looks like this. So these balls represent an amino acid. And then if I kept going and I said serine, and then I wanted to keep adding and building a protein, okay, so now we're eventually building this really large molecule, okay, and this large molecule will be called a polypeptide. It's called a polypeptide because poly, which stands for many, and peptide, because think about that peptide bond that forms. There are many peptide bonds that form, so it's a polypeptide molecule. And it's usually a short string of amino acids. It can also be a large string of amino acids, but in essence, it's a string of amino acids. And eventually, this polypeptide can become a protein, a functioning protein. When we think of proteins, we might be thinking of maybe this meat. And when we think of proteins, you should think about like the actual substance that makes us who we are. We have to consume protein, and we digest the protein that we eat. We break apart that protein, and we extract the amino acids from the protein we consume. Those amino acids are essential for us because we can take those amino acids and then build our own proteins. So the growing polypeptide will be a protein eventually. And what is a protein? A protein is a functioning macromolecule that can go on to do various functions in our own body. And they can be very sensitive to the environment depending on the conditions. Now I'd like to take a look at the structure of proteins and look at this in more detail. So the structure of proteins fall under 4 different types, and they progressively get more complex as we move down. So the very first type of structure that we see with proteins is what's called the primary structure. The primary structure simply means the basic arrangement of these amino acids. It's a linear arrangement of amino acids, and it tells us just the order of amino acids. It doesn't tell us anything about how these amino acids interact with each other. It's just telling us the basic linear order of these amino acids. The next structure of protein is more complex, and this is when we begin to see the side chains interacting with each other. And what can happen in the secondary structure is we see 2 different types structures forming as a result of the side chains interacting with each other. The first one is what's called beta pleated sheets, and it kind of looks like this jaggedy, like pleated sheet goes up and down, almost if you were to fold your paper, like so that you'd get like this jagged line of up, and down. So it has like this pleated look to it. And the other type of structure that you would get for secondary structure is what's called an alpha helix. So going back to perhaps your review, if you ever thought about DNA acting as a double helix, how does a helix look like? Well, in essence, it pretty much looks like the curl of a hair, and it kind of spirals downward. And here it's that same concept that has the same structure. Alpha helices do the same thing, where it kind of spirals and it curls down. So structures, secondary structures, can do both of these things. The next structure is the tertiary structure. So it gets a little more complex as we move down. And the tertiary structure now is when we begin to see these secondary structures interacting with each other. So this is a great example showing the alpha helices that look like curls and the beta pleated sheet. There's a lot going on with the tertiary structure. What I want you to understand is that the side chains that come with the amino acids are extremely complex, and they each have a very specific property, and they can each interact with another kind of amino acid in a specific way. And so what we end up getting is this really complicated structure that is dependent on how the amino acids are arranged. And so this type of arrangement of these amino acids will depend on how the folding of the protein takes place. This is important because structure equals function in a protein. So a protein has to have a particular structure for it to function in a particular manner. Now the last type of structure of a protein is what's called a quaternary structure. Not all proteins have a quaternary structure, but many of them do. And that's really what the quaternary structure is referring to, is when you have multiple separate polypeptides now coming together and interacting with each other to form one protein. So that sums up proteins, and to be continued as you move on to the next macromolecule.